Hello, today we'll be studying a very important topic related to the cell, which is the cell organelles. The first cell organelles which I'm going to talk about is the mitochondria. Mitochondria was first observed by Altman in 1894 and he named it as bioblast. But later on, Branda in 1897 named it as mitochondria. This mitochondria looks like a bean or a rod. Some people also call it as rod shape or tube-like structure. This mitochondria, when it is present inside the cell, have the capacity to change its shape. They can fuse with one another or split into two, which means if they are together, if this is a one mitochondria, it can split into two different mitochondria and become two. Now how this can happen? If you can see here, matrix is present. This matrix divides itself and due to which this mitochondria splits into two. Another thing which is very important here is, as they are capable of changing its shape, therefore their number, length, shape and interconnectivity always changes. There is no fixed connectivity. Sometimes mitochondria remains associated with microtubules. The microtubules are present inside the cell, so they remain associated with them. Now let's talk about the structure of mitochondria. There are two membranes present in the mitochondria. One is the outer membrane. The outer membrane is smooth and plain, as you can see here, followed by inner membrane. Inner membrane is folded and it is longer than the outer membrane, as it is too much folding is present. This folding is called as cristae. This folding and the inner membrane consist of small granule like structure which is called as F1 particle. As you can see here, it is not present only in one place of the inner membrane. It is present all over the inner membrane. I've just showed in one place. The place between the outer membrane and the inner membrane is called as intermembrane space. In the inner membrane you will find a matrix present. Inside this matrix there is DNA of mitochondria and ribosome of mitochondria. These are very important parts of mitochondria. So let's start with the parts of mitochondria. The first is the membrane. As I've already told you that there are two types of membrane. One is the outer membrane and the other one is the inner membrane. The first, outer membrane, it is composed of approximately 50% of lipid. Because it occupies almost 50% of the weight. Okay, and it has lots of enzyme, a mixture of enzyme present in the outer membrane. What is the role of the enzyme? Main role of this enzyme is to degrade tryptophan. It degrades tryptophan. What is tryptophan? Tryptophan is nothing but the amino acid. And another important feature of this enzyme is that it elongates fatty acid. These are few important things which is the main role of enzyme. Apart from this, there is one more thing which is present, is that if you see the structure of the mitochondria and the outer membrane, it is something like this. This is the outer membrane. So this outer membrane consists of group of proteins. Okay, places, there are many places where there are group of protein. So this group of protein come together to form a porins. What is the role of this porin? This porin acts as a pore. Okay, and this pore helps in the permeability of the cell. and this porine helps in the permeability of the membrane which is present here, and it helps in the easy intake and uptake of the molecules inside the outer membrane. But these molecules which enters the outer membrane cannot enter the inner membrane because inner membrane doesn't have such pores. Now we'll start with the inner membrane. Inner membrane is again very important. It is subdivided. This inner membrane is subdivided into two. One is the inner domain, this inner domain is called as inner boundary membrane. This inner boundary membrane is nothing but present just next to the outer membrane. As you can see here, these are the inner boundary membrane. Followed by this inner boundary membrane, there is another domain which is called as the crista. This crista is nothing but the folding, which are like this. So this is the inner membrane. This inner membrane is longer than the outer membrane because it is highly folded. Crista. Crista is again very important because this is something which gives the folding to the inner membrane, and apart from this, it is very important to increase the surface area of the inner membrane. This crista consists of one very important particle attached to it, which is called as F1 particle. This F1 particle is present not only in crista but also in the inner membrane. The full inner membrane is covered with the F1 particle. This inner membrane consists of this F1 particle, and this F1 particle looks like this. It consists of a head, pedicel and base. What is the main role of this F1 particle? The main role is that this is the place where the ATP is synthesized. This is the only place. Now how this ATP is synthesized? What happens is that this F1 particle squeezes ADP plus PI and gives out ATP. This is all about the crista. The next one is the matrix. It is a gel-like material called as matrix, and it is highly viscous. It is more viscous than the cytoplasm. Why it is more viscous than cytoplasm? Because matrix consists of less amount of water. Therefore it is viscous. What do you mean by viscous? Thick and sticky. It is very thick and sticky, and therefore it is viscous more than the cytoplasm. This is all about the mitochondria. Thank you.